Hi, today the question is regulation of purine nucleotide metabolism, right? Regulation of purine nucleotide metabolism. Before going to the metabolism, the end product of purine metabolism is uric acid. So you should know the normal level of uric acid. Usually it is 2 to 5 in milligram per deciliter in males and 4 to 7 milligram per deciliter in females. Okay, this level should be maintained. This is blood level of uric acid. If the level is high means the condition is called hyperuricemia. The level is low means you know the condition ADA, adenosine deaminase disorder, right? So you should maintain the purine nucleotide metabolism. Regulation means controlling the pathway, that is metabolic pathway, according to the need of the cell. The end product will be synthesized, right? If it is low means the pathway will get synthesized. If it is high means it should be stopped. That is a control point. Otherwise, increased means disease, decrease also produces diseases. That is why regulation of purine metabolism is very, very important. Next one is, we have 4 types of regulation. That is, number 1, availability of the substrate. For any metabolic pathway, initial product is very, very important. So, availability of the substrate. The substrate availability is high means metabolic pathway also high and end product also high. Availability is less means pathway also will be less. Second one is controlling enzymes or regulatory enzymes that is present in the same pathway, right? This is enzymes present in same pathway, metabolic pathway. Third one is hormonal regulations. Which hormone either induces the pathway or decreases the pathway. The last one is allosteric modifiers. These products actually compounds, they will actually increase the pathway or decrease the pathway depending upon the situation. Allosteric activators, either modifiers, that is inhibitors or stimulators. These are all the 4 regular topics for your regulation of any metabolic pathway. Okay. Coming to purine synthesis, this is controlled by 2 mechanisms. One is actually concentration of the PRPP, phosphoribosyl pyrophosphate. Second one is feedback regulation. These 2 are very, very important points for your regulation of purine metabolism. That means if PRPP increase means increase the purine nucleotide pathway. Vice versa, if the PRPP level is low means purine metabolic pathway will be decreased. This PRPP concentration depends on the rate of its 3 points. One is synthesis, second one is how it is utilization, third is degradation, how it will get degraded. In these 3, the synthesis part is very, very important because again this depends on the availability of the ribose 5 phosphate. First product, very, very important product is ribose 5 phosphate. You know very well, this is synthesized from HMP shunt pathway in carbohydrate metabolism, right? Ribose 5 phosphate. Second one is activity of the PRPP synthase. This is the first enzyme which is controlling the first step of purine metabolism, PRPP synthase. So second one is feedback inhibition. You know the meaning of feedback inhibition. That is, the final product of the pathway will either inhibit or stimulate the pathway depending upon the situation. Okay, this is known as feedback inhibition. That is, it will stop the previous enzymes or previous steps in the metabolic pathway depending upon the situation, that is substrate availability. So the first 2 steps of purine metabolism is very, very essential for your regulation of purine metabolism. The first step is otherwise known as rate limiting step, that is conversion of ribose 5 phosphate into PRPP. For this conversion, the enzyme is PRPP synthase. Actually, the other name of this one is ribose phosphate pyrophosphokinase. So this will be automatically utilized 1 molecule of ATP which will get converted into AMP. Here the cofactor for this reaction is magnesium ions. All these points are very, very important, right? So this is a rate limiting step, first step of purine synthesis. Actually in your some books says activation of the purine metabolic pathway, first step. Some books says the routine step number 1 like that. Second one is, that is preparatory step, some book given. Second step is The glutamine combines with this PRPP, produces glutamate and phosphoribosylamine. This step is also very important. For this conversion, the enzyme name is PRPP amidotransferase. This amidotransferase is inhibited by IMP, GMP and AMP. You know the meaning of IMP, GMP and AMP. Guanosine monophosphate, adenosine monophosphate, inosine monophosphate. These are the allosteric modifiers which inhibit these enzymes or stimulate these enzymes, step 1 and step 2 enzymes, to regulate the purine nucleotide pathway. You got it? In the purine metabolic pathway, you should draw the diagram or draw the first 2 steps for the regulation of purine metabolism, your 5-mark question or essay question, right? The next one is, we have the intermittent product AMP, GMP and IMP. This AMP and GMP regulate the formation of IMP, because you know the IMP only synthesize GMP and AMP, right? So with the help of, we need 2 enzymes for this conversion, adenosine synthase and IMP dehydrogenase. These 2 enzymes are very, very important for the regulation of purine metabolism, right? Adenosine synthase and IMP dehydrogenase, which requires actually 1 molecule of GTP and 1 molecule of ATP, right? This AMP and GMP again inhibit the enzymes in salvage pathway. You know the meaning of salvage pathway. This is actually de novo synthesis of purine nucleotide, main pathway. Emergency pathway is called as salvage pathway. For the salvage pathway, enzyme name is HGPRTase. This enzyme is also inhibited by this product, that is IMP, AMP and GMP. So AMP and GMP inhibit the enzyme HGPRTase, which converts hypoxanthine to IMP and guanine to guanosine monophosphate GMP. This is the third point. Last point, in this way, both de novo synthesis of purine metabolism and salvage pathway are balanced. If de novo synthesis occurs, means there is no salvage pathway. If salvage pathway occurs, means de novo synthesis is not coming. So to provide purine nucleotides to the brain like organs, the continuous supply is very, very essential. So that is why the main pathway is purine nucleotide de novo synthesis. The emergency pathway is salvage pathway. So to maintain the purine nucleotide metabolism, this AMP and GMP will maintain the balance, that is either inhibit the salvage pathway and stimulate the main metabolic purine nucleotide de novo synthesis, or if there is no de novo synthesis of purine metabolism, it stimulates the salvage pathway to provide nucleotides to the brain like organs. Okay, bye.